Hello children, I'm Ravichandra from Sri Chaitanya, an organization committed in students' dreams of achieving something big. And stay with me for an hour. I'll take you to a tour of a chapter called Photosynthesis. And I promise you, you'll be extremely happy after listening to it. Don't leave me, all right? So we're going to start this chapter, Photosynthesis, and we'll run through this topic very fast. First, I start with the definition of photosynthesis. It is the synthesis of organic compounds. What is that organic compound? Glucose from water and CO2, from simple inorganic compounds. Next important point, which time it takes place. Light phase occur during daytime. Even the dark phase also occur during daytime. This is something which you study in your lower classes. What are the cells in which photosynthesis occur? Green cells. This is a simple point. What type of conversion? It's an energy transduction process. Light energy is converted to chemical energy. First ATP in light phase, then this is used in the synthesis of glucose. Now, what is this process? This process is anabolic process, constructive. And what is the cell organelle involved in this process? It is chloroplast. Chromatophores are seen in blue-green algae. They are the extensions of plasma membrane which contain pigments. Right. Then, next one is the equation for photosynthesis. 6CO2 plus 12H2O give rise to glucose, oxygen, and water. Look here. Water is donating its hydrogens to carbon dioxide. So it means that water is losing hydrogens, therefore it's getting oxidized. Carbon dioxide is gaining hydrogens, therefore it's getting reduced. So the compound which gets oxidized is water, therefore it's a reducing agent. And the compound which gets reduced is carbon dioxide. Having said that, in some green sulfur bacteria, it is not water, it is hydrogen sulfide which is actually getting oxidized. That is the reason you know pretty well it is anoxygenic photosynthesis. Then the most important one, from where these compounds? Now look at this, this is simple. Carbon in glucose should from carbon dioxide. Hydrogen in glucose should come from water. Oxygen in glucose come from carbon dioxide. Oxygen liberated come from water. And hydrogen here should come from water. And oxygen here will come from carbon dioxide. Well, these lines are looking a little difficult for you to understand, isn't it? Shall I make it easy? 12 oxygen atoms are there. 6 2s are 12. It means that all the oxygen liberated is coming from water, isn't it? Then the oxygen present here and the oxygen present here should definitely come from carbon dioxide. That's it. Same thing I've written here. What did I write here? Carbon in glucose come from carbon dioxide. Hydrogen in glucose come from water. Oxygen in glucose come from carbon dioxide. Oxygen released is from water, and the scientist's name you remember, Neil. Hydrogen in product water come from substrate water. Oxygen in product water come from carbon dioxide. Nice, we made a good start. Right, where is photosynthesis observed? It is observed in monorans, protistans, and plants. And what is about monorans? It shows both oxygenic and anoxygenic. Oxygenic in blue-green algae, whereas in green sulfur and purple-green sulfur, it is anoxygenic. That's what I written. Photosynthesis is oxygenic in monorans, that is blue-green algae is an example for that. Protistans and plants. Photosynthesis is anoxygenic because sulfur compounds is going to get oxidized. That is the reason it is anoxygenic. This word is little tricky. Chemoautotroph. The general belief of most of you is autotroph means it should perform photosynthesis. But the fact of the matter is, even though they are autotrophs, they doesn't perform photosynthesis. They oxidize the inorganic chemicals present in them and thereby gain energy. So therefore, they are not examples of photosynthetic organisms, even though they are autotrophs. Next one. Photosynthesis is a physicochemical process. Why we say that? There are some physical processes and chemical processes involved in this. What are the physical changes? Here I've written absorption of light is physical, excitation of electron from chlorophyll is physical, photolysis of water is physical. Whereas synthesis of ATP and NADPH2, Synthesis of sugar from glucose, these are the ones which are chemical. That is the reason your textbook says it's a physico-chemical process. Photosynthesis is an ultimate source of food, naturally, because it is going to synthesize glucose, which is the starter, producers, and thereby the entire food chain depends on it. And photosynthesis not only release oxygen during light phase, it also produces glucose during dark phase. These are the two vital compounds which are produced. That completes the introduction part to photosynthesis, children. Now we get into some experiments which are there in your textbook. The first is variegated leaf. Look at this. A leaf has got both green and non-green parts. Only green parts respond to iodine test, starch iodine test. Non-green parts doesn't. So it means that it explains the role of chlorophyll in photosynthesis, that is variegated. Then black paper leaf experiment. Let us not waste our time here. A part of the leaf is covered with a black paper. The remaining part of leaf was normal. That part of the leaf which is not covered with black paper respond to iodine test, not the one which covered with black paper. So therefore this experiment explains the role of light in photosynthesis. Then Moll's half leaf experiment. Half of the leaf is exposed to KOH. And you know, children, KOH absorbs all the CO2. So therefore this part of the leaf doesn't get CO2. So therefore there is no starch. The other part will get CO2, there is starch. So therefore the role of carbon dioxide in photosynthesis is explained by Moll's half leaf experiment. Now the next important one is the scientist names. The first in that list is Priestley. Let me show you a nice diagram. Based on that, we'll be discussing on it. Look at this. In the first bell jar, you observe the candle is burning and the rat is happy. Isn't it? Yes. But after some time, the candle was put off and the rat died. You may ask me why such sad things happen. Simply because there is no oxygen. The deficiency of oxygen is the reason why the rat got suffocated and the candle was put off. Then you know what Priestley did? He thought that he should not kill rat. So therefore what he kept? He kept a mint plant under the bell jar. Now, mint plant is able to perform photosynthesis. And you know, children, in photosynthesis, CO2 is actually taken in and oxygen is liberated. So the burning candle and mouse was respiring and liberating CO2. And the plant is taking that CO2 and by a process called photosynthesis evolved oxygen. And that oxygen kept the candle burning and kept the mouse happy or alive. So that is the most important point. So, and what Priestley said here, what I have written here, experiment 1, candle and mouse in bell jar. The candle did not burn and the mouse died. Both candle and mouse damaged the air, all right, by taking up all the oxygen. Then what the next one? Experiment 2. Here the candle, mouse and mint plant. All right, what did you observe? The candle continued to burn in closed space and the mouse was alive. The plants restore the quality of the air. What did Priestley said? Priestley said plants take bad air, that is CO2, and liberate good air, that is oxygen. That's very simple. And there is one word. Is there any way where we can lit the candle without opening the bell jar? All right, there are many ways where we can lit the candle without opening the bell jar, using the burning glass or magnifying glass, better way of saying, using a mirror, all right, to direct light into the jar, are some of the ways to relight the candle. This is a question which was asked. Then Ingenhousz, you know what did he do? He did the Priestley's experiment. He kept the bell jar, the entire setup during daytime. Because light was there, the plant was able to perform photosynthesis and evolve oxygen, keeping the mint plant, keeping the mouse alive. In the same setup, when it kept in dark, you know what happened? There is no photosynthesis, there is no evolution of oxygen, and the entire mouse and candle was put off. Not only that, children, Ingenhousz did one more experiment. Oxygen bubbles were seen from a plant called hydrilla. The name is not in your book, hydrilla. And this oxygen bubbles evolved only during daytime because light phase and dark phase occur only during daytime. In the night time, there was no oxygen bubbles from hydrilla. So again, what is the ultimate summary of Ingenhousz experiment? Light is essential for photosynthesis. That's a very important point. Sacs, plants produce starch or glucose. All right, in green substances in plants are located in special bodies called chloroplasts. And you know what is that green substance? The green substance is chlorophyll. Then we talk about Engelmann. Just like Spider-Man, He-Man, Engelmann is associated with action. Now, what did he do? He took a light, prism to break light, an alga called cladophora, and an aerobic bacteria. Let me show you the diagram, what happened here. Based on that, we'll go through this. Look here. What did the prism do? Divided the light into 7 colors. He exposed cladophora to all these 7 colors. That wavelength at which the rate of photosynthesis is more is blue and red. Naturally, oxygen is evolved as a product in light phase, you know pretty well. Because of oxygen evolution in large quantities in blue and red, more aerobic bacteria accumulated in that suspension, clearly signifying that blue light and red light are the most effective light in photosynthesis. And be careful, children. Engelmann is associated with action spectrum, not absorption spectrum. And what is an alga used by him? Cladophora. What is an alga used by Calvin in his experiments? Chlorella. So for these 2 examples are important. Alga used by Engelmann is cladophora, by Calvin is chlorella. And we talk about Warneel. This is simple experiment. All of you know that. All right, we already discussed that. If it is water, all right, then the oxygen is evolved. And if it is not water, if it is H2S, oxygen is not evolved. Look at this 2. Look at these 2. Carbon dioxide is common in both. Only when water is there, oxygen is evolved. When water is not there, H2S is there, there is no evolution. That was a time where the people was little confused about this oxygen release. Is it coming from CO2 or water? Because both of them contain oxygen. Warneel, conveniently with this experiment, working on green sulfur bacteria, proved to the world that oxygen evolved come from water, not from carbon dioxide. This is a very important point. Reuben and Carmen, not that much important, children. If you look at what did he do? He has taken O16 here. He took O18 here. So naturally, this will be O16, this will be O16, and this will be O18. So it's very clear that oxygen is coming from water, therefore it is O18. Oxygen in glucose and the product water is coming from carbon dioxide, therefore it is O16. And by the way, let me put one question. What are the 2 isotopes used in photosynthesis? In light phase, it is O18, and in dark phase by Calvin, it is, yes, you guessed correctly, C14. All right. Then after that, all the scientists' names I kept here, children. Priestley, mint plant, Ingenhouse, hydrilla. Engelmann, cladophora, green sulfur bacteria. Calvin, chlorella. Hatch and Slack, sugarcane. Right. Then we talk about the chloroplast. Now, this is something which you already heard in a chapter in your first year. Chloroplast contain membrane envelope. Outer membrane is more permeable than inner membrane. The space is called periplastidial space. Stroma contain proteinaceous fluid. It has got many circular DNAs, unlike mitochondria, which has got one circular DNA. It contains all 3 types of RNA, not all 3 types of RNA polymerase. All 3 types of RNA polymerase is there inside the nucleus. Such kind of questions, you should be careful, children. 70S ribosomes produce proteins. Enzymes of dark phase. Your textbook says carbon reactions are synonym for dark phase. Remember that. Reduction of carbon dioxide to sugar occur in stroma. So shall I say that both proteins and sugars are produced in stroma of chloroplast? Yes. Enzymes work only in intact chloroplast. If I isolate all the chloroplast, put it in a storehouse and can produce glucose by exposing them to light? Absolutely not. The enzymes turn volatile on isolation. Isolated chloroplast can perform light phase but not dark phase. Remember that. This is the diagram of chloroplast. All the thylakoids group together to form grana. All right, so look here, this is the thylakoid. And these are all the thylakoids. This is said to be one granum. The space inside that is said to be the lumen. And this thylakoid, which joins the two adjacent granum, is said to be the stroma lamellae or stroma thylakoid. This is said to be the membrane system present inside the chloroplast. All right, grana, a part of membrane system, it's a stack of thylakoids. Grana represent a group of granum is called grana. Pigments are present on grana. Stroma lamellae, just now I have shown you. All right, the two adjacent grana are joined by stroma lamellae. And you know, this is very important. On this children, on this grana, there is both photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. Whereas on stroma lamellae, there is only photosystem 1. Now, what is photosystem? It's a group of pigments. All right, here you see only photosystem 1, whereas in these two, you see both photosystem 1 and 2. Remember, I repeat again, grana thylakoids contain both photosystem 1 and 2, whereas stroma lamellae will have only PS1. That is the reason non-cyclic electron transport is not seen there. These are the points which you know it. Grana both PS1 and PS2, stroma only PS1. Thylakoid membrane is energy transducing membrane because it is impermeable to protons. Allow the proton accumulation on one side, create a proton gradient. When you break this gradient, ATP is synthesized. Therefore, we say energy transducing membrane. Could you answer? In mitochondria, what is the energy transducing membrane? Yes, inner membrane of mitochondria is energy transducing membrane. What is the significance of light phase? There is a synthesis of ATP and NADPH2. Good question. There is a division of labor within the chloroplast, isn't it, children? Light phase help in the synthesis of ATP and NADPH2, and dark phase job is to utilize this ATP and NADPH2 to synthesize glucose. So I'm going to give you two statements. Tell me which is correct. Light phase depends, dark phase depends on light phase. Statement number 2, dark phase depends on light. Which is correct? Dark phase depend on light phase is correct. Dark phase depends on light is not totally correct because for dark phase no light is needed. But dark phase depends on the products of light phase. That is an important point which you have to remember. Now this is a very one area where you normally make mistakes. These are called tangential, these are called longitudinal walls or radial walls. When the light intensity is low, the chloroplasts are arranged with the flat surfaces facing light because they want to absorb more light. So therefore, when the light intensity is less, how are the chloroplasts? Chloroplasts are perpendicular to these transverse walls, perpendicular to these longitudinal walls. Perpendicular, I repeat again, when the light intensity is less, chloroplasts are present perpendicular to these longitudinal walls or radial walls or parallel to the transverse walls. This is correct. One more way. When the light intensity is high, look at the chloroplast. They are present parallel to the longitudinal or radial walls and they are perpendicular to the transverse walls. So it means that the chloroplasts keep moving. They are dynamic. Depending on the light intensity, they tend to move from one orientation to another. Different shades of green color is because of chlorophyll A, chlorophyll B, carotenoids and xanthophylls. So chlorophyll A is blue-green, chlorophyll B is yellow-green, carotenoids is yellow-orange, xanthophylls are yellow. Have you seen old and mature leaves, children? Yes, they are yellow because of a lutein, a xanthophyle. Then carotenoid, strictly speaking, carotenoid is a combination of carotene and xanthophyle. What is the color of xanthophyle? Yellow. What is the color of carotene? Orange. So therefore, yellow-orange. Easy way to remember. And chlorophyll A is blue-green and chlorophyll B is yellow-green. Remember this? All these four pigments can be separated by a technique called paper chromatography. Now let us talk a little bit about chlorophyll pigments. It's a lipid, remember that? It is fat soluble because it's a lipid. It is water insoluble. And it is seen in Monera. In blue-green alga, you see chlorophyll. Whereas in some sulfur bacteria, you see bacteriochlorophyll. It is also a type of chlorophyll, but it is not truly chlorophyll A. All right, Monera, Protista, and plants. They contain, what is this? Chlorophyll, blue-green alga, chlorophyll A only. Diatoms and dinoflagellates, A and C, you know that. Euglenoids, A and B. Now we go to fungi, no chlorophyll. Now we go to plants. Green alga, A and B. Brown alga, A and C. Red alga, A and D. So start in higher plants, it is A and B. So what about blue-green alga? Only A. So remember that. Those are the pigments which I've kept here. Euglenoids, green alga, brown alga, red alga. Alga is very unique in having all types of chlorophyll pigments. Higher plants, A and B. The head of chlorophyll, now if you look at this, it looks like a tennis racket. The head of chlorophyll is called porphyrin head, and the tail is said to be phytol tail. And this head contains 4 rings. And these 4 rings are said to be pyrrole rings. The central element in these 4 rings is magnesium. It is joined to nitrogen present in these 4 pyrrole rings. So this is porphyrin head, and this is phytol tail. Just one point you remember, magnesium is present in the center. Chlorophyll A is a chief pigment. All right. Chlorophyll A has absorption peaks in blue and red. Look here. This is called absorption spectrum. Observe here. In blue light and red light, chlorophyll A is absorbing large amount of wavelengths in blue and red. And in other wavelengths, you could see the absorption of that is very less. Blue and red, absorption is more. What about chlorophyll B? Same. Blue and red, absorption is more. But if you look at the peak, children, chlorophyll B absorbs more blue when compared to A. Likewise, chlorophyll A absorbs more red when compared to chlorophyll B. I repeat again, B absorbs more blue when compared to A, and chlorophyll A absorbs more red when compared to B, but both of them absorbs more of blue and red. This is absorption spectrum, a graph which explains about different wavelengths of light absorbed by pigments. What about carotenoids? In the graph which is given, it is blue and green. It shows absorption peaks in blue and green, carotenoids. You remember that also. I'm going to show you those graphs. Carotenoids, children, they are lipids, just like chlorophyll. Just like chlorophyll, they are fat soluble. Just like chlorophyll, they are water insoluble. Carotenoids are yellow-orange. Absorption peaks, this is the point I was referring to, blue and green. They are accessory pigments. Which one? Carotenoids and xanthophylls are accessory pigments. I'm going to show you what are those accessory pigments. They absorb light and transmit light to the reaction center, and they protect the reaction center from getting photo-oxidized. So these are the 2 jobs of accessory pigments. Both carotenoids and xanthophylls does this job. Now let us talk about xanthophylls. Just like carotenoids, they are lipids, they are fat soluble, they are water insoluble. The color I told you is yellow. Lutein, I was talking about a yellow xanthophyll which is present in old mature leaves. Is it an accessory pigment? Yes, and it helps in wide range of light to be absorbed. Even carotenoids, it protects the reaction center from high intensities of light. This is the point which I was referring to. Brown alga is brown because of a xanthophyll called fucoxanthin. This is an outside chance. Phycobilins. They are water-soluble proteins. This is interesting. Unlike chlorophyll and carotenoids, which are water-insoluble fats, phycobilins are water-soluble protein. The study of alga is phycology. So therefore, phycobilins are seen only in alga. Phycoerythrin, phycocyanin are the 2 types of pigments which are present in alga. Then we talk about action spectrum. We again talk about Engelmann. He for the first time reported a graph which explains the rate of photosynthesis at different wavelengths of light. All right. Now look here, children. This is it. Observe this. How do you know the rate of photosynthesis at different wavelengths of light? By the amount of product formed. What is the product which is liberated into the atmosphere? Oxygen. So by looking at how much amount of oxygen is liberated, one can easily tell the rate of photosynthesis. And look here, it's very clear that in blue light and red light, the rate of photosynthesis is more. We already discussed that. All right, action spectrum peaks are there in blue and red. Then, this is a very interesting graph. Did you observe that in your textbook, children? Yes. Now observe here. This is the black colored one is the action spectrum, which is the contribution of all the pigments. You should not say action spectrum of chlorophyll A. There is nothing like action spectrum of chlorophyll A, understand? And this blue colored one is the absorption spectrum of chlorophyll A. What did you observe? I'll tell you three things. One, action spectrum and absorption spectrum of chlorophyll A are overlapping at the peaks. Yes. What does it mean? It means that whichever wavelength of light chlorophyll A absorbs in large quantities, at the same wavelength, rate of photosynthesis is also more. It means that chlorophyll A is the chief, is the headmaster. Yes or no? Then, one more important point. Chlorophyll A and absorption spectrum of chlorophyll A and action spectrum are not exact one-to-one overlap. No doubt they are overlapping at peaks, but it is not exact one-to-one overlap. What does it mean? It tells that other pigments also have a role to play. Other pigments, look here, a captain is an important person in a team. Whenever captain plays, the team normally wins. Normally, not always, isn't it? Same is the case here. Chlorophyll A is no doubt chief, but doesn't mean that chlorophyll A alone controls the entire photosynthetic rate. It is not the case. Look at this. There is very less absorption of light by chlorophyll at this wavelength. But look at rate of photosynthesis. It is not dropping to zero. What does it mean? Other pigments are there which holds and allows photosynthesis to take place. So telling that peaks overlap to some extent is right. Telling that it is not exact one-to-one overlap is also right. I'm pretty sure that you understood this. This is the point which I have written here. Chlorophyll A absorbs blue 420. Nice number. All right, and red strongly, and these very wavelength also produce maximum photosynthetic activity. All right, that means that the peaks overlap. Action spectrum of photosynthesis and absorption spectrum of chlorophyll A is not exact one-to-one overlap. Photosynthesis is not driven by chlorophyll A alone. It depends on the combined absorption of chlorophyll A, accessory pigments, or one more way of saying accessory pigments broaden the action spectrum beyond the absorption spectrum of chlorophyll A. Nicely I've written here. This is a very important point to remember. Then we come to a concept of photosystem. Already I was talking about what is the meaning of photosystem. Photosystem is a cluster of pigments. It's a group of pigments, all right, involved in photosynthesis. Now, what does a photosystem include? Photosystem include a reaction center, a light harvesting complex, primary electron acceptor, and some proteins and enzymes. All these four put together is said to be a photosystem. Reaction center, children, always a chlorophyll A molecule. Light harvesting complex or antenna complex include not only chlorophyll A, but chlorophyll B, carotenoids, and xanthophylls. But when it comes to reaction center, it is chlorophyll A. Your textbook says except one molecule of chlorophyll A. They are referring to that is reaction center. The reaction center contains a special chlorophyll A. Actually, it's a pair, but in your textbook they gave only one special chlorophyll A molecule capable of converting light energy to chemical energy. The light harvesting complex or antenna complex include all four pigments, but these three are said to be accessory pigments, all right, because they absorb light and transmit light to the reaction center. All right, the accessory pigments include chlorophyll B, carotenoids, and xanthophylls. What is the function of accessory pigments? It absorbs light and transmits light to the reaction center, protect the reaction center from excessive light intensity. This is a diagram which explains you the concept of photosystem. This is the entire photosystem. It contains chlorophyll A. Here I have shown two, but take it as one chlorophyll A molecule. This is light harvesting complex which absorb light and transmit light to the reaction center. And this is the primary electron acceptor, which is also a part of photosystem. And you also see what is called the entire process taking place, even in the presence of some proteins. So all these four put together, you can call it as photosystem. This is not that much important. In resonance energy transfer, excited, look here, if this is the pigment, its electrons are getting excited, de-excitation. This energy is transported to the pigment number 2. All right, so therefore in this way, so not only pigment directly absorbs light from outside, but also some amount of energy is also transported from the previous pigment. So this is how the transfer of energy occurs from one pigment to another. All right. To naturally, when the energy is lost, wavelength is going to what is called increase. So it is going to transfer to a pigment with higher wavelength, is something which you know pretty well. Now we talk about photosystem 1 and photosystem 2. First photosystem to function in non-cyclic is PS2. Even though your textbook says both the photosystems operate simultaneously, but photosystem 2 operate little earlier than photosystem 1 because it excites earlier. Discovery, it is second. That is the reason why you call it as 2. Reaction center is P680. Absorbs maximum light at 680. Some of you are thinking that it absorbs only at 680. No. Absorption maxima is 680. Normally it absorbs from around 650 to 680. All right, chlorophyll A, that is reaction center I'm talking about. So absorption maxima is 680. That is the reason why it is said to be P680. It is located on grana thylakoid membranes only. PS2 is absent on stroma lamellae. Photosystem 1, it is the second photosystem to function. It is discovered first. Reaction center is P700. It absorbs maximum light of 700 nanometers. It is located both on grana thylakoids and also on stroma lamellae. This is interesting. PS1 is seen not only on stroma lamellae but also on the grana thylakoid. Now we are in the last part of light phase, where we look at how the light phase operate. All right, now listen properly to what all I'm talking here. If at all you get a feeling that I went a little fast, pause the video and see it one more time. Surely you'll understand this. All right. So how we are going to talk about. Let me explain you this, children, on a white page. Let me take a white page and explain you. That will make your job more easier. Isn't it? Look here. What we do here, children, this topic, I'll divide into 2 or 3 installments. This non-cyclic electron transport. We'll study in 3 installments such that it becomes more easier for you to follow. Right, look here. This is a thylakoid membrane. Observe, thylakoid membrane. This is the thylakoid membrane, and the space inside the thylakoid membrane is said to be the lumen. Now listen carefully. All right, concentrate more. This is the thylakoid membrane, and the space inside that is said to be the lumen. And this entire thing is stroma. So shall I put my hand like this and write all the 3 parts? Yes. Now what are all the 3 parts what are right here? This is said to be stroma. Yes. And this is said to be the thylakoid membrane. Yes. And this is said to be the lumen. Up to here, no problem. Stroma. This is the thylakoid membrane. And the space inside the thylakoid membrane is lumen. S means stroma, thylakoid membrane, and lumen. Up to here, there is absolutely no problem. Now, what I do is, let me change the pen. Right, now I'll take a black one. So let me see if you can properly see it, because on the white background, black looks better. Now, let us start with, on the thylakoid membrane, you know pretty well, on the thylakoid membrane, you see the presence of pigments. Just now I told you, it contains photosystems. So therefore, this is a photosystem. And the reaction center is P680. The reaction center is P680. Correct. And there are other pigments also. Yes. And they are accessory pigments. So shall I tell you a small equation? Photosystem is equal to reaction center plus light harvesting complex. Reaction center plus light harvesting complex. Light harvesting complex is also called antenna complex. And light harvesting complex or antenna complex include chlorophyll A, chlorophyll B, carotenoids, and xanthophylls. I exclude chlorophyll A. The remaining 3 are said to be accessory pigments. I repeat again. Photosystem or pigment system is equal to reaction center plus light harvesting complex. Now I break light harvesting complex into 4. A, B, carotenoid, xanthophyle. I exclude A from that, and the remaining 3 are said to be accessory pigments. Perfect. All right. Now it is going to absorb the light, children, in the form of photons. What will happen now? When it absorbs light in the form of photons, electrons get excited. And now it becomes P680. I'm going to show you like this. This is an electron which is getting excited. Look here, I am P680. When light hits me, and also thanks to all the pigments which absorb light and transfer that light energy to me, so my electrons get excited. This is how it is. This is the high energy electron. And what did you study? High energy means more unstable, isn't it, children? So this P680 star is an excited form of this P680 with high energy electron. So what does it do? This is going to lose, look here, this is going to lose its 2 electrons or 1 electron, whatever may be. It's going to lose 2 electrons to a compound called pheophytin. This name is not given in your textbook. They said primary electron acceptor. They did not say what is that primary electron acceptor. This is pheophytin. No problem. I'm P680. When light hits me, this is P680 star here. Then I'm going to donate the 2 electrons to pheophytin. Now, after donating 2 electrons to pheophytin, now the P680 star, let me show you like this, for convenience only for convenience. Now it becomes P680 plus plus. Why? Because it has lost 2 electrons, naturally. Now P680 plus plus, all right, now wants the 2 electrons and it asks pheophytin, can you give me 2 electrons? Pheophytin says, nothing doing, I'm not going to give. Then P680 plus plus gets worried. I'm deficient of 2 electrons. Who is going to help me? Water says, I will help you. So therefore, this is going to help in the breakdown of water into 2 electrons, 2 protons, half O2, provided if it is 1 water. If it is 2 water, 4 electrons, 4 protons, and 1 molecule of oxygen. All right? Yes. Now, this 2 electrons goes to P680 plus plus such that it can become P680. Now you understood. P680 plus light, P680 star. P680 star becomes P680 plus after losing 2 electrons. P680 plus after gaining 2 electrons from water again becomes P680. If somebody asks you which photosystem is responsible for breakdown of water, your answer should be PS2. I forgot to write you, this is PS2. It operates first, I told you no. This is photosystem 2. This photosystem is responsible. One more question. Where does this breakdown of water occur? This breakdown of water occur on the lumen side. Lumen side. Is there any protein playing a big role here? There is a protein. This protein is oxygen evolving complex, which is attached to the thylakoid membrane but towards the lumen side, which is playing a good role in this process. Everybody is happy. P680 is happy because it got 2 electrons, and pheophytin is happy because it got 2 electrons from this. Up to here, there is absolutely no problem. What it happens here, children, is there is loss of 2 protons here. And this 2 protons is going to increase the proton presence in lumen. What did you study in chemistry? More protons means less pH or acidic. So the lumen starts becoming more and more acidic because of loss of protons from water. This is called photolysis of water. Manganese and chlorine are the 2 important elements which are required for photolysis of water. I'm going to stop here and go back and see what did I write, all right, with regard to this particular topic. Right, now look here. Let me see what I've written here. Chalo, from here. The first step in non-cyclic electron transport is absorption of light by photosystem 2. Light excites P680 of the reaction center. This is totally correct. I'll change the pen to red. All right. Then, what the next one? It is an uphill transport. Yes, children. P680 to P680 star is surely an uphill because this has more energy when compared to this. It's an uphill transport. P680 lose an electron and become oxidized. P680 star lose an electron and become oxidized to P680 plus. The excited electron is then transferred to primary electron acceptor called pheophytin. Photolysis is a light-driven process, splitting of water into protons, electrons, and oxygen. Where does it occur? On the lumen side. How many photosystems are involved? Only one, PS2. These electrons lost from P680 must be replaced continuously. So water acts as electron donor. Water gives electrons to P680 such that it becomes this. Oxygen evolving complex is involved. Minerals, manganese, and chlorine. All right, provide electrons to this, add more protons to lumen, decreasing the pH. All right, so nice. We have reached up to here. This is the first part of non-cyclic electron transport. I'm pretty sure that you understood this without any problem. Then comes the next very important point, all right, which I'm pretty sure you have to concentrate here, children. What will happen next? Listen here. The two electrons from here. Shall I change the pen? Yes, I will do. All right. Now, it is going to lose the two electrons. The two electrons are lost from pheophytin to a compound called plastoquinone. This is a very important compound. It is going to lose two electrons to plastoquinone. Plastoquinone is a lipid andi. So this is thylakoid membrane. What did you study in cytology? All right, you studied that thylakoid membrane has contained lipids, phospholipids. All right, any membrane will have lipids and proteins, just like plasma membrane. So the thylakoid membrane contains lipids. Plastoquinone is also a lipid. You are a lipid, I am a lipid, you can go. So plastoquinone can freely move, hydrophobic. It can freely move within the lipid bilayer. So it's mobile, it can move easily. Nobody is going to ask plastoquinone where you're going, why you're going. So plastoquinone is always easy to move. Otherwise, this thylakoid membrane is impermeable. Be careful. Impermeable. But plastoquinone is an exception. It can move without any problem. Did all of you understood this? Chalo. It is accepting two electrons. All right. Now, not only it accepts two electrons, it accepts two protons also. From where? From the stroma. Then naturally, by accepting two electrons and two protons, plastoquinone is going to become plastoquinol. In organic chemistry, you studied ons on reduction give rise to all. That's very simple. Now, by accepting two electrons and two protons, I am plastoquinone. I accept two electrons from pheophytin. I accept two protons from stroma. Now I have got two electrons and two protons. I am free to move. So therefore, what I do? I move from that side. Look here. This is the membrane. Okay. I move from that side. From the stroma side, I pick. This is from stroma side. I move towards that side, towards the lumen side. Chalo, I'm going to move up to here. I'm going to go up to here. So this is PQH2, plastoquinol. Why H2? Because it's accepting two electrons and two protons, therefore PQH2. So I'm moving from here to here. Who am I? Plastoquinone. Chalo, what do I do later? After reaching here, I'm going to stand like that. On which side I am? I'm here, on the lumen side. On the membrane only, but on the lumen side. Why I'm standing? I'll tell you the reason. All right, I will wait. I will wait for what? I will wait for a compound called cytochrome B6F. What does cytochrome B6F do? It is going to wake up, and what does it do? Immediately, it is going to pull two electrons only electrons from PQH2. Once it pulls the two electrons from PQH2, its two protons now are more free to move. And these two protons will be dumped into the lumen. This is interesting. I repeat again. I am PQ, present towards the stroma side. I accept two electrons thanks to pheophytin. I accept two protons from stroma. Now I become PQH2. Now I tend to move on this membrane towards the lumen side. I'm free to move because the lipids are permeable. I'm a lipid. Phospholipids are there in thylakoid membrane permeable. After reaching here, I lose two protons and I lose two electrons. Who is responsible for that? Cytochrome B6F. It says that I'll pull the 2 electrons from you, and it pulls 2 electrons. Once it pulls the 2 electrons, the 2 protons are lost. What did you observe? 2 protons are lost from dash to dash. 2 protons are lost from stroma to lumen. Question is, how are these 2 protons moving? These 2 protons are moving through plastoquinone. With the help of plastoquinone, the 2 protons are moving. Question you're going to ask me is plastoquinone acting as a proton channel? No. Does it act as a proton pump? No. Then how can it carry proton? Physically, with its hands. It is physically carrying it. Simple way of saying, well, in a lighter way I said, but actually it is oxidation reduction. It's a chemical process which is actually carrying protons. So protons are carried chemically. All right, not in the form of proton channel, not in the form of facilitated diffusion, not in the form of active transport, not even in the form of simple diffusion. Then how? I'm getting reduced here and I'm getting oxidized here. That's it. It is a reduction oxidation. So after getting reduced here, I have hydrogens. I go here, I drop the hydrogens, means protons, and then again come back here. So it means that it is a chemical process which is actually transporting protons from stroma to lumen via plastoquinone. Who is the man of the match here for carrying protons from stroma to lumen? Difficult, very difficult, because there are 2 very fierce competitors. One is definitely plastoquinone, which is physically carrying protons, simple way, proton carrier or hydrogen carrier, because this is hydrogens. And one more competitor is cytochrome B6F. By pulling the 2 electrons from PQH2, it is allowing the 2 protons to move into the lumen. If you ask me, both of them are joint winners. Let us be fair. But we'll say one important statement. PQ means plastoquinone is a proton carrier. No problem there. Proton carrier. It is moving from here to here. And cytochrome B6F is proton pump. Right word. Why proton pump? Because it is actually helpful in pumping protons into the lumen. Pump? Some of you got it out? You said pump? Yes, I said. All right. Then pump is a word used. You know, water is pumped from lower to higher. So pump is a word used from lower to higher. Do you mean that there are low protons here and high protons here? Of course, high protons are here. Because protons are constantly added by breakdown of water. So there are more protons here. So protons are moving from low to high. Against the concentration. That is the reason we require some energy. Protons have to move from low to high against the concentration. That is the reason we require energy. Not only that, when there are more positive charge inside, look here, this mischievous positive charge protons doesn't want more protons to move, to join. Once you get a seat in a bus, you want the bus to move. You don't want the other passengers to come. All right, same is the case here. Once the protons are here, they don't want the remaining positive charge protons to move here. So if at all you want to transport protons from here to here, you have to do it with force, because there is a lot of resistance here. So not only there is a difference in concentration, there is also an electrochemical gradient which is between here and here. Isn't it charge difference? Concentration difference. Why concentration difference? Less protons, more protons. Why charge? Less positive charge, more positive charge, and protons have to move to an area of more positive charge. Protons are also positively charged. That is the reason this movement is said to be active. Means, better way of saying, it is against the concentration gradient. It requires energy. Now the question you're going to ask me is, who is going to provide the energy? Very simple. The downhill movement of electrons, listen carefully, the downhill movement of electrons is responsible for liberating energy. Now you understood what is the job of this chlorophyll molecule. The job of this chlorophyll molecule, electrons get excited, and its electrons getting de-excited, higher energy to lower energy. From higher energy to lower energy, energy get liberated. You may ask me what I should do with this energy. Come on, help and transport protons from stroma to lumen. This is easy to answer. Your textbook also talks about the concept of redox potentials. Let me not talk too much of chemistry here, but I'll just touch and go here, this topic, redox potentials. Look here. The meaning of redox potential is the ability to get reduced, potential to get reduced. Look here. When P680 star is going to pheophytin, electrons move from pheophytin to plastoquinone from here to here. It means that electrons are moving from lower redox potential value containing compound to higher. Look here, children, if I want to pull something, all right, from the previous one, I should have a higher redox potential. I should have more reduction potential, then only I can pull. Isn't it? Yes. So all these electron carriers are kept in the increasing order of redox potentials. So let me give you, imagine it is minus 20 here. All right. So that is pheophytin. Then imagine this is 0 here, or maybe I put it as minus 10 here for convenience, or this is 0 here. What is it? Is it not increasing order of redox potentials? Yes, because minus 10 is more than minus 20. Agreed? If I take a difference of redox potentials, Sunday, listen carefully, difference of redox potentials, minus 10 minus minus 20. It comes to plus 10. Now you understood the difference of redox potentials is positive. And what did you study in chemistry? The difference of redox potential, if it is positive, free energy will be negative. Yes or no? And you know that when the free energy is negative, it is energy releasing process. That is the reason energy is released in this downhill movement of electrons. So in two ways I explain. In the language of higher energy of electron to lower energy, when the electron moves from its higher energy to lower energy, energy is liberated. One more, electrons are going to move from lower redox potential to a compound of higher redox potential. Difference of redox potential is positive, means free energy is negative, means it's an exothermic reaction, energy releasing process. Ultimately, if somebody asks you, what is the utility of this energy? Utility of this energy is for the transport of protons from stroma to lumen. It is taking a little time, but I want you to understand this concept. Of course, you understood this concept. I want you to remember this concept thoroughly. Your tutor have explained all this nicely. This job is to read this topic such a way that there is no need for you to read for more amount of time in the coming days. Look here, what did I write here? What did I write beyond this? We were up to here, we read water. Now let me talk about plastoquinone and cytochrome B6F here. What did I write here? I kept 1 or 2 white pages. All right, pheophytin. It is the primary electron acceptor, correct? It is present on the stroma side of thylakoid, correct? All right. Then plastoquinone, very important. It is a lipid. It is hydrophobic. It can easily pass through the lipid bilayer. It is a mobile hydrogen carrier because it accepts electrons and it accepts protons. Therefore, hydrogen. It is mobile electron carrier. Moves from stroma side to the lumen side. It accepts electrons from pheophytin. It accepts protons from stroma side. PQ becomes PQH2. It moves towards the lumen side. It loses 2 electrons thanks to cytochrome B6F, which pulls the 2 electrons. In this process, the 2 protons are added to the lumen. All right, protons are added not only by PQ, but also by water. Now, look here, what did I write? Protons, PQ does not form a pore or a channel for protein transfer. It transfers protons from stroma to lumen by binding them chemically and carrying them as a part of PQH2. Totally clear. Why energy is needed to transport protons from stroma to lumen? Because of concentration difference and electrical gradient. Here I've written, movement of protons from lumen is against the concentration gradient, as high protons already exist in the lumen, and further accumulation is energetically uphill. And added to that, when more protons enter into the lumen, the lumen becomes electrically positively charged related to stroma. Positive charges repel other positive charges. Therefore, incoming protons are electrostatically repelled. So, energy is needed to force more protons. Now you understood why energy is needed to carry protons from stroma to lumen. And from where this energy come from? Already I told you, it is the downhill movement of electron or difference of redox potentials. Light excites electron in PS2. These electrons now possess high free energy to low free energy. Hence, electrons lose free energy. It helps in uphill transport of protons. In non-cyclic electron transport delta E, the difference of the redox potential is positive because electrons move from a carrier with lower or more negative redox potential to one with higher or more positive redox potential. If delta E is positive, then the delta G, the free energy, will be negative. Negative delta G means the free energy decreases, hence energy is released spontaneously. And let me talk about the cytochrome B6F complex. It helps in proton accumulation inside the lumen. PQ gets oxidized. 2 protons get liberated into the lumen. 2 electrons from PQ is accepted by cytochrome B6F. And proton carrier is plastoquinone. Proton pump, these 2 points are important. Proton carrier is plastoquinone. Proton pump is cytochrome B6F. Both of them work together for the transport of protons from stroma to lumen. Cytochromes are iron-containing proteins, and cytochrome B6F oxidize PQH2, and it is going to reduce plastocyanin. So the next electrons from cytochrome B6F, children, electrons from cytochrome B6F reach plastocyanin. So let me show you again. This is electrons. This is P680 star, then to pheophytin, then to plastoquinone. Yes, plastoquinone not only accepts 2 electrons but also 2 protons. I'm sorry. PQH2. PQH2 loses 2 protons into lumen. Then it loses 2 electrons to cytochrome B6F. From here, the 2 electrons goes to plastocyanin. So this entire movement is downhill. Higher energy of electron to lower energy. Lower redox potential to higher redox potential. Higher energy to lower energy, energy get liberated, the difference. Higher redox potential to lower redox potential to higher redox potential, the difference of redox potential is positive. Therefore, the free energy will be negative. Energy is liberated. Both this energy liberation will help in the movement of protons from low to high. Now you understood why this electron transport should occur. Plastocyanin, hemocyanin, gastropods, blood pigment, blue. Yes. So it means that plastocyanin is a copper-containing electron carrier. So why I brought hemocyanin? Understand that. Just like that, copper, here also copper in that way. So it's not that related to that. All right, cytochrome, it oxidized cytochrome B6F. Is it not pulling the 2 electrons from cytochrome B6F? Means oxidizing cytochrome B6F and reducing P700 is going to give the 2 electrons to P700 plus. It occur on the lumen side. Electron carrier is mobile. Electron energy from pheophytin to plastocyanin. From pheophytin to plastocyanin, higher to energy, lower. Redox potential from pheophytin, lower to higher. This is uphill transport. And P680 star to plastocyanin is downhill. These points are very important to remember. I'm pretty sure that you understood this entire process. Let me show you the diagram, children, with regard to this that will help you to understand. This is photosystem 2. Again, all of you know this is photosystem 2, P700. Again, it is going to lose, it becomes P700 star, then it becomes ferredoxin reducing substance, then to ferredoxin, then to NADP. This is how it moves. All right. So again, when P700 star loses 2 electrons, it becomes P700 plus plus. Now the plastocyanin is going to give its 2 electrons to P700 plus plus. This is what is called the photosystem 2, which is operating. P700 star, ferredoxin reducing, ferredoxin, and ultimately to NADP plus. It absorbs, reaction center is P700, discovery first, absorption maxima at 700, located on grana thylakoids and also on stroma lamellae. P700 to P700, this is uphill, to NADP plus is downhill. P700 donates electrons to ferredoxin reducing substance, then accepts electrons from plastocyanin. Ferredoxin reducing substance is an iron sulfur protein. Ferredoxin ultimately donates electrons to NADP, an enzyme, remember, FNR, ferredoxin NADP reductase, is an enzyme responsible for this. Location of FNR is on the stroma side. Look here, this is the entire diagram you could see here. This is the one we studied. Water is breaking up here. All right. First step is P680. It is losing electrons. Pheophytin is somewhere here. All right. Then it loses electrons to plastoquinone. We already discussed. It is accepting protons. Then PQH2. It is losing 2 protons and losing 2 electrons to cytochrome B6. From here to plastocyanin. From plastocyanin, the electron goes to PS1 to ferredoxin and ultimately to NADP. So evolution of oxygen is on the lumen side. More proton accumulation is on the lumen side. Breakdown of water is on the lumen side. Synthesis of NADPH2 is on the stroma side. All right. And this FNR enzyme is there on the stroma side. Already, just this is very simple. Now, more protons got accumulated. More protons got accumulated in the lumen, isn't it? Up to here, no problem. Now, what will happen? These protons which are there in the lumen will tend to move towards the stroma. Why? Come on. When there are more protons, system always will be from more to less. All right. When more protons are there, they want to tend to move to an area where it is less. But this membrane, this thylakoid membrane, is impermeable to protons. That is the reason there is one carrier. This time, it is acting as a facilitated diffusion carrier. This tail is acting as a channel for the movement of protons. And when protons move from higher to lower, this is from lower to higher, energy is needed. When the same protons move from higher to lower, energy will be liberated. And that energy liberated will help in the synthesis of ATP. This is said to be chemiosmosis. Here I'm going to explain you. Does that scheme, of course, include all this? PS2 to primary electron acceptor, primary electron acceptor to PS1, PS1 to primary electron acceptor to NADP+. Products of non-cyclic electron transport is ATP, NADPH2 and oxygen. And cyclic electron transport, children, let me show you here. Cyclic electron transport occur, all right, under conditions where a photosystem is exposed to a wavelength greater than 680, or when there is a deficiency of NADP+, or when there is ATP synthesis needed but not NADPH2. Cyclic can occur both on granular lamellae and always on stroma lamellae because there is no PS2 there. So in cyclic, photosystem loses electrons to ferredoxin. From ferredoxin, the electrons go back to plastoquinone, cytochrome B6F, plastocyanin, and again go back to the same photosystem. That is the reason why I say it's cyclic. And in the cyclic electron transport, there is no FNR involvement, there is no NADPH2, there is no PS2, therefore there is no photolysis of water and there is no evolution of oxygen. Only protons get accumulated in the lumen. When the same protons move to this coupling factor, ATP is synthesized. So the common product formed both in cyclic and non-cyclic is ATP. And the last part in light phase, chemiosmosis, scientist's name is Peter Mitchell. Its synthesis of ATP due to the breakdown of proton gradient. It is applicable for both chloroplast and mitochondria. What we require in chemiosmosis? We require an impermeable membrane. We require a proton gradient across the membrane. We require a proton carrier from stroma to lumen. We require a proton pump. And we also require a proton carrier from lumen to stroma. All these are needed for chemiosmosis. We already discussed all that. Why there are more protons? Because of photolysis of water and active transport. Why there are less protons in stroma? Why there are less protons? There is a reduction of NADP and there is active transport. Look here, children. First I create a gradient. This is the thylakoid membrane. First I create a gradient. Very, very less and very, very high. Once more protons get accumulated in the lumen, now on one fine morning, I'll carry these protons from high to low. Just like in hydroelectric power, from higher height, when the water is dropped, the turbines move, energy is liberated. When a ball is rolled from a higher height, energy is liberated, the potential energy. Yes or no? So the difference. So same way, from when the protons move from higher to lower through this coupling factor, when protons move from higher to lower, energy is liberated. And that energy is responsible for ATP synthesis. This is called photophosphorylation. First, I accumulate protons in the lumen thanks to water and plastoquinone. Then after some time, I carry these protons from lumen to stroma through coupling factor. The tail part of coupling factor acts as a proton channel, and because of this the head part is going to rotate because energy is liberated. This is called proton motive force, and that rotation, conformational change, will help in the synthesis of ATP. This is the entire concept of ATP synthesis. And CF0, CF1, this is the CF0 and this is CF1. So the CF0 acts as a proton channel. When protons move from high to low, energy gets liberated and ATP synthesized in the head part. This entire thing is said to be ATP synthase. The tail is CF0, embedded in thylakoid membrane. Head is CF1, it is on the stroma side. CF0 help in facilitated diffusion channel for proton. CF1 use this proton motive force to generate ATP. This is a nice diagram. This is CF0 and this is CF1 phase. Here we see the production of glucose. Then what we did in light phase is to synthesize ATP and NADPH2 because they are needed for dark phase. So we produce all the raw materials needed. Now our job is to see the glucose synthesis. Without wasting any time, we'll get into it. It is called carbon pathway. We already discussed about this. It is temperature dependent because enzymes are involved. Enzymes are temperature sensitive. In enzymes chapter you studied. It is dependent on the products of light phase, not on light. Products of light phase. And they are high energy intermediates. Why you say intermediates? Because ATP and ADPH2 are surely intermediates because ultimately they produce glucose. All right, they are intermediates, and that high energy intermediates we synthesized in light phase. It is not dependent on light. Now there is a proof. Immediately after light becomes unavailable, dark phase occur for some time till ATP and NADPH exhaust. So it means that it is not dependent on light. It is dependent on light phase. All right. Then occurrence. It occurs in stroma of mesophyll cells mostly. So Calvin children, a scientist name, he got Nobel Prize in 1961. Experimental plant is chlorella. Isotope used is C14. Sodium bicarbonate is the form of radioactive carbon which is injected into chlorella, C14. Chemical used to kill algal cells is hot methanol. What this man did is he added radioactive carbon in the form of sodium bicarbonate into algal cells. He allowed photosynthesis to take place for a certain period of time. He dropped it in hot methanol. Photosynthesis stopped. By using autoradiography, he was able to know the intermediate compounds. Again, he took these algal suspensions. Again, he did the same. This time he increased the time of photosynthesis. In this way, he deciphered the entire Calvin cycle. In 1961, along with Basham, he got a Nobel Prize. Technique used to know the intermediate compounds is autoradiography, and Benson and Basham are his associates. And other contributions of Calvin, light energy is converted to chemical energy during light phase, and enhanced solar energy research as renewable source. All right, now children, there are two pathways of carbon dioxide fixation. Your textbook clearly says about that. One is C3 pathway, a glucose is synthesized, and one more. In C4 pathway, also a glucose is synthesized. So we'll start with the C3 pathway, and the initial carbon dioxide acceptor is RUBP, the first stable product is PGA. And in C4 pathway, it is PEP's initial carbon dioxide acceptor, and the first stable product is oxaloacetic acid. Let us start, all right, this Calvin cycle, and let us see one by one. Now listen carefully. So we'll start this Calvin cycle and produce glucose. Let us see. 6 molecules of RUBP. 6 molecules of RUBP, ribulose biphosphate. It's a 5-carbon compound. I take 6 molecules of RUBP. Okay. Then I add 6 molecules of carbon dioxide and 6 molecules of water. And what is produced is 12 molecules of phosphoglyceric acid, which is a 3-carbon compound. This is a very easy one. 6 molecules of RUBP combined with 6 molecules of carbon dioxide and 6 molecules of water to form 12 molecules of RUBP. And listen here, RUBP is a 5-carbon compound. It's a keto sugar. Now I'm adding 1 carbon in the form of carbon dioxide. Then naturally it forms a 6-carbon compound, isn't it? It's going to be highly unstable. It breaks into 2 3-carbon compounds. And this 2 3-carbon compounds is PGA. So easy to remember. So it's very clear that RUBP is a 5-carbon plus 1 6-carbon. It breaks into 2 3-carbons. So 1 RUBP plus 1 CO2 produce 2 PGA. So 6 RUBP plus 6 CO2 will produce 12 PGA. What is this step called? This step is called carboxylation phase. What is an enzyme responsible for this? The enzyme responsible for that is Rubisco. How many ATP utilized in this process? 0. How many NADPH2 utilized in this process? 0. Let us see all the points with regard to carboxylation phase. All the points with regard to carboxylation phase. It is the fast and the crucial phase, yes. Initial carbon dioxide acceptor is a 5-carbon keto sugar, yes. First stable product is phosphoglyceric acid, which is a 3-carbon non-sugar, yes. Enzyme is RUBP carboxylase. It has oxygenase activity as well, but here it is not acting as oxygenase. Number of Calvin cycles needed to burn glucose is 6, because 1 CO2 is said to be 1 Calvin cycle. So 6 Calvin cycles has to operate in order to produce glucose. This is the equation, children. 6 RUBP plus 6 CO2 and 6 water give rise to 12 PGA. Substrate is keto sugar. Product is phosphoglyceric acid. No ATP used, no NADPH2 used. Perfect. That completes the first phase of Calvin cycle, which is said to be carboxylation phase. Now, let us go to the next phase, children. What will happen in the next phase? Let us see what we do in the next phase. Look here what we did. We took 6 molecules of RUBP, yes, and we added 6 molecules of carbon dioxide, and we produced 12 molecules of phosphoglyceric acid. Let us do some calculations. 5 6 are 30, 1 6 are 6, 36, 12 3 are 36. Easy to remember, absolutely no problem. This completes the next phase, that is said to be the carboxylation phase. Now let us talk about the next phase, reduction phase. What I do? These 12 molecules of PGA, listen here, 12 molecules of PGA, I convert to 12 molecules of phosphoglyceraldehyde. What did you study in your chemistry? Acids is becoming aldehyde. When can acid become aldehyde? Only when you reduce it, reduction. So therefore, reduction means what? Addition of hydrogens. So what I do is, here I add 12 molecules of ATP and 12 molecules of NADPH2. That's it. Look here, children. In PGA, there are 6 CO2. This 6 CO2 present in PGA To that 6 CO2 present in PGA, I'm adding hydrogens thanks to NADPH2, which is giving hydrogens to it. So 12 PGA contain this 6 CO2. To this 6 CO2, I'm adding hydrogen. COH. CHO. That is nothing but PGAL. It's a sugar. It's a triose sugar, 3-carbon sugar. It's a monosaccharide, also called glyceroldehyde phosphate. And no prizes for guessing. What is it called? This is rightly called reduction phase because you are reducing this compound to this. It is not a single step process, it's a many step process, but in your textbook they have given in a short form, reduction phase. Let us read what is there under reduction phase. What I have written under reduction phase. All right, let us see. Reduction phase is the next step. I'll change the pen. All right, reduction phase. Please concentrate here. 12 PGA molecules are converted to 12 PGAL. In this, 12 ATP and 12 NADPH2 are needed. All right, ATP needed for 6 CO2 in the reduction phase is 12. Correct, 12. ATP needed for 1 CO2 in the reduction phase will be 2. As simple as it. Same way, NADPH for 6 CO2 is 12 and for 1 CO2 it is 2. All right, and what the first form triose sugar? The first form triose sugar is PGAL. And how many PGL molecules we produce, children? We go back. How many PGL molecules we produced? 12. You know what I do with this 12 PGL? All right, I break it into 2. 10 PGL and 2 PGL, okay? Now what I do with this 2 PGL? This 2 PGL, I produce 1 molecule of glucose. Naturally, because this is 3-carbon compound, 2 3s are 6, 1 6. Yes, I'm very happy that I produce glucose. So out of 12 PGL, I take 2 PGL and produce glucose. You got a doubt. Why can't you take all 12 and produce 6 glucose? Why you're taking only 2? There is a logic here. Now, each PGL is a 3-carbon compound. Yes. 10 PGL means 30 carbons. Yes. These 30 carbons are utilized in the regeneration of 6-RUBP. 10 3s are 30, 5 6 are 30. So now you understood. 10 PGL, first we have to keep it aside. Then only we can produce 6-RUBP. If you can't regenerate RUBP, you can't continue this cycle for long. That is the reason why we took only 2 PGL. So this is the reduction phase, children. And this is said to be the regeneration phase, where 10 PGL are utilized in the regeneration of 6-RUBP. And I want to tell you, here I use 6 molecules of ATP. So it's very clear. Let us count how many ATP we produce utilize in total. 12 plus 6, 18 ATP. And how many NADPH2 we utilized? 12 NADPH2. So 18 ATP and 12 NADPH2 are needed. For how many carbons? 6 CO2. For how many glucose? 1 glucose. So for 6 CO2, 6 Calvin cycles, 1 glucose, we require 18 ATP and 12 NADPH2. 1 Calvin cycle, or for 1 CO2, we require 3 ATP and 2 NADPH2. In the reduction phase, number of ATP, 12, and in the regeneration phase, 6. For 1 CO2, in the reduction phase, 2, in the regeneration phase, 1. Total 3. So let us just see what we have written, all right, with regard to reduction phase and regeneration phase, children. We already went up to here. Yeah. 2 PGL out of 12 are utilized to produce glucose, understood. 10 out of 12 PGL, regeneration, you understood that. PGL formed in Calvin cycle help in the synthesis of glucose, sucrose, and ultimately starch, you know it. Substrate for reduction phase, PGA, it's not a sugar. Product is PGL, it's a sugar. ATP is 12 in the reduction phase, 12 in the regeneration phase, NADPH2. For 1 CO2, 2 and 2. Then we talk about the regeneration phase. Already I told you 10 PGL. I utilize 6 ATP to produce 6 RUBP because 10 3s are 30, 5 6 are 30. Regeneration of RUBP is needed for continuity of this cycle. ATP needed is 6, but 1 ATP is needed, all right, for 1 CO2. Substrate in the regeneration phase is PGL. Product is RUBP. Is it true that both substrate and product are sugars? All right, ATP used is 6 and NADPH2 used is none. So look at this cycle here I have written. 6 CO2 plus 6 RUBP, 12 PGA, carboxylation phase. 12 PGA, 12 ATP and 12 NADPH2, 12 PGL, reduction phase. 2 is utilized in the production of glucose and the remaining 10 PGL are utilized in the regeneration of 6 RUBP, very nicely given. 6 ATP are utilized here. This is a repetition, children. 6 CO2, this much. 1 CO2 is this much. 6 CO2 in carboxylation phase, 0. 1 CO2 in carboxylation phase, naturally be 0. 6 CO2 in the reduction phase, 12. 1 CO2 in the reduction phase, 2. If they ask you reduction of CO2, 1 CO2, say 3. Reduction phase means say 2. So be careful about this. 6 CO2 in the regeneration phase, 6. 1 CO2 in the regeneration phase is 1. Important statements about C3 plants. They are mostly in temperate regions. Yes, C3 plants are mostly temperate. Temperature optimized 20 to 25 degrees. They work because temperate regions, children, the temperature optimized less, temperature is less. Common example, rice, wheat, potato, tomato, bell pepper. All right, and tomato. We come across this, all right, in factors. Initial carbon dioxide, RUBP. First stable product is PGA. It occurs in the stroma of the chloroplast of mesophyll. Starch is synthesized in stroma of chloroplast. Rubisco act as both carboxylase and oxygenase. Location is in mesophyll cells. Is PEP carboxylase observed in C3 plants? Yes. Is photorespiration seen? Yes. I'll tell you what is photorespiration. All right. So these points you'll understand after some time. Effect of you'll understand after some time. Next, carbon dioxide saturation point is around 450 ppm. Now what is this? 450 parts per million means for every 10 power 6 air molecules, CO2 at 450 units for every 10 power 6 air molecules is CO2. So at 450 is the amount of CO2 present for every 10 power 6 molecules of air. Then maximum photosynthesis is reached in a C3 plant. But atmosphere contain 0.03%, means only around 350 ppm. Atmosphere contain 350 ppm, saturation is at 450 ppm. Use a common sense. It means that present level of carbon dioxide is surely less than what is needed for saturation. That is the reason you grow the C3 plants in carbon dioxide enriched environment. Those are called greenhouse crops. Tomato and bell pepper will come across that. All right, many greenhouse crops. Then Kranz anatomy is absent. Chloroplast dimorphism is absent. Number of carboxylations required is 6. No decarboxylations. And number of cell types is only 1. Nice. It took a little time for me to get rid of Calvin cycle, but we discussed it perfectly. All right, next time there should not be any confusion. Before I get into Hatch-Slack cycle, first of all, you have to understand the birth of Hatch-Slack cycle is for what sake? Why Hatch-Slack cycle has to operate in some plants? So for that, we should touch the concept of photorespiration first. So first we'll get rid of photorespiration, children, and then we'll come back to this, the concept of Hatch-Slack cycle. So let us see what is photorespiration. Let us see what is photorespiration just for a minute. This is the place where we talk about photorespiration. Let me explain you this concept of photorespiration first. Very simple. Hardly takes 2 minutes. Now listen carefully. Imagine I have RUBP molecule, yes. Now this time imagine oxygen is more, not carbon dioxide. Interesting. Previously RUBP combined with CO2. Enzyme name was Rubisco. Now I'm Rubisco. I have got two active sites, one for RUBP, one for either oxygen or CO2. When oxygen concentration is more, oxygen will run and bind this place and competitively inhibit the binding of CO2. So therefore, I can say this, high amount of oxygen act as a competitive inhibitor of RUBP carboxylase activity, because carboxylase activity cannot occur when oxygen bind here. Oxygenase activity is seen. That's it. Now listen carefully. RUBP is going to combine with oxygen because atmosphere contain more amount of oxygen when compared to CO2. Then this will occur. What did you study in your chemistry, children? Any compound in the presence of oxygen has to break, isn't it? Yes. So therefore, RUBP is a 5-carbon compound. It breaks up into a 3-carbon compound called phosphoglyceric acid and a 2-carbon compound called phosphoglycolic acid. Phosphoglycolic acid. A 2-carbon compound called phosphoglycolic acid. These are the two products formed. 3 plus 2, 5. This is 5. Yes or no? Yes. Up to here, no problem. Now, are you going to permit me to take 2 molecules of RUBP in place of 1? Okay, no problem. 2. Then naturally, then how many PGA formed? 2 phosphoglyceric acid. How many phosphoglycolic acid formed? 2. Yes. Now listen, because 3 to the 6, 2 to the 4, 6 plus 4, 10, 5 to the 10, no problem. Enzyme name RUBP oxygenase. Now listen carefully. These 2 molecules of phosphoglycolic acid, look what happens. This is going to pass through a series of reactions, and form 1 molecule of phosphoglyceric acid. Passing through chloroplast, peroxisome, mitochondria. These are the 3 cell organelles. Now let us count small 2 to the 4. This is phosphoglyceric acid 3. Means there is a loss of dash carbon dioxide. There is also a loss of dash water. Just like in respiration, substrate. Just like in respiration, substrate plus oxygen. Just like in respiration, produce water and CO2. Yes or no? But unlike in respiration, there is no ATP synthesis. There is no NADPH2 synthesis. Unlike in dark phase, there is no sugar formation. No ATP, no NADPH2, no sugar. I repeat again, 5 carbon, 2 molecules, 10, 2 3 is 6, 2 2 is 4. These 2 molecules of phosphoglycolic acid, 2 to the 4, 1 CO2 is lost and the remaining 3 becomes 1 molecule of phosphoglyceric acid. Now use all your common sense and tell how many molecules of phosphoglyceric acid is formed. Let us see. For 2 molecules, 2 molecules here, from here 1, 3 molecules. So the number of phosphoglyceric acid molecules formed is 3. Why we are counting? Because ultimately this PGA should form what? This PGA should form PGAL and should form glucose by Calvin cycle. Keep this in mind. How many PGA molecules formed in RUBP oxygenation? Total 3. And what about RUBP if it's get carboxylated, children? How many PGA formed? For 2 molecules. For 6 molecules of RUBP, it is 12. For 2 molecules of RUBP, it should be 4. Now you got this. So 2 molecules of RUBP, when it combines with 2 molecules of carbon dioxide, that is Calvin cycle, the number of PGA formed is 4. 4 PGA, 4 PGL, glucose. Then, if the same 2 molecules of RUBP combines with oxygen, the number of PGA formed is 2 plus 1, 3 only. So, less PGA, less PGL and less glucose. That is the reason why I say photorespiration is an unproductive process. Shall I tell you one more reason why it's unproductive? There is a loss of carbon in the form of carbon dioxide. Added to that, in this entire process, there is utilization of ATP and NADPH2 to make things more worse. That is the reason photorespiration is not a useful process. And who is the villain here? Who should be blamed for photorespiration? Please don't blame oxygen for that. It is the enzyme Rubisco which has a dual nature. It is providing seat for CO2. All right, that's good. But it can also provide seat for oxygen if oxygen concentration is more than CO2. So it is the Rubisco site which is causing a problem for us. If Rubisco says, I absorb only CO2, I don't take oxygen, then we wouldn't have seen this entire process. The fact that this is going to absorb even oxygen, means take even oxygen, catalyze even RUBP oxygenation, that is the problem. But if Rubisco is the problem, shall we send Rubisco out? No, don't do that. Because without Rubisco and without Calvin cycle, we can never produce glucose. The only way we can produce glucose is by using Rubisco and other enzymes of Calvin cycle. Neither we can keep Rubisco with us because quite often it is getting oxygenated. Neither we can throw Rubisco out because if we throw Rubisco out, there is no way we can produce glucose. This is a very tricky situation, isn't it, children? Yes. Then 3 plants, sugarcane, maize, sorghum. Shall I say SMS? Ha. Sugarcane, maize and sorghum. Thought a lot for a long period of time. They came out with a solution. You know what is the solution? They ask Rubisco and all the other enzymes of Calvin cycle, can you please change your place from mesophyll to bundle sheath? Then the enzymes ask why we should change our place. Because bundle sheath cells are large, they are more green because there are more chloroplasts, and more importantly, they are properly insulated with suberin, so you feel without any disturbance you can stay there. All the enzymes of Calvin cycle which are normally present in mesophyll cells, on the request of sugarcane, maize and sorghum, they all pack their bags and they go to bundle sheath cells. This is interesting. All right, bundle sheath cells. Now let us see, after all the Calvin cycle enzymes going to bundle sheath cells in sugarcane, maize and sorghum, how is the glucose produced and how is photorespiration prevented there? This is the last topic for us to discuss. Concentrate what all I am saying here. All right, this is the photo. Before we go to the the C4 pathway, children, before we go to C4 pathway, let us go through this photorespiration, what I have written. Photorespiration is a light-dependent process in which RUBP oxygenation occurs, leading to consumption of oxygen and release of CO2 without production of ATP or sugar. It's a very marathon statement. I kept too many points there. Shall I make it, break it and make it easy? All right, time, daytime, yes. Light, high light intensities. Only in high light intensities, there will be more oxygen release. Only when there is more oxygen release, RUBP oxygenation occur. CO2 concentration is less, oxygen concentration should be more. And this occur only in C3 plants because I told you sugarcane, maize and sorghum have found out some way, we don't know, but found out some way to avoid photorespiration by asking all the Calvin cycle enzymes to go to bundle sheath cells. We'll be talking about that story in 2 minutes. Cell organelles for this, chloroplast, mitochondria and peroxisome. It is not useful. Rubisco act as oxygenase. High concentration of oxygen reduces the carboxylase activity. This is the competitive inhibition. 1 RUBP, 1 oxygen, this is a 5 carbon compound breaking into a 3 carbon and 2 carbon. Enzyme name oxygenase, it occurs in chloroplast. Conditions, high light, high oxygen and low CO2. Same thing I take, children, for 2 molecules. All right, then 2 molecules of PGA and 2 molecules of phosphoglycolate or phosphoglycolic acid. What will happen to these 2 molecules of phosphoglycolic acid? It forms 2 molecules of glycine in peroxisome, 1 molecule of serine in mitochondria, ultimately 1 molecule of phosphoglyceric acid. So it's very clear that these 2 molecules of phosphoglycolic acid, all right, is going to ultimately form 1 molecule of phosphoglyceric acid. So now count 2 PGA here, 1 PGA. So total 3 PGA molecules are there for 2 molecules of RUBP. So the number of PGA formed will be 3. What is utilized in photorespiration? ATP and NADH. What is not produced in photorespiration? Sugars, ATP, and not produced. Why photorespiration is a wasteful process? For every 2 molecules of RUBP undergoing normal carboxylation, all right, the number of PGA formed is 4. When the same 2 molecules of RUBP undergo oxygenation during photorespiration, the number of PGA formed is only 3. Only 3 molecules of PGA are effectively recovered. 1 carbon is lost as CO2, children. We have already seen. Less PGA, less glucose. Added to that, ATP and NADPH2 are consumed. No ATP is produced, no sugars. All right. Now at this point, don't talk about C4. Look at C3. CO2 fixation rate and high light intensities. We'll discuss this after we discuss C4. Children, is photorespiration okay? Yes. Now we get into this C4 pathway. We stopped our story by saying that all the Calvin cycle enzymes are asked to enter into bundle sheath cells by SMS plants. Then what it will do in mesophyll? What it will do in mesophyll? Let us now look. In mesophyll, it keeps 6 molecules of phosphoenol pyruvic acid. This is a 3-carbon compound. Yes or no? No RUBP. 6 molecules of phosphoenol pyruvic acid. Then it is going to combine with 6 molecules of carbon dioxide. Then it forms 6 molecules of oxaloacetic acid. It is a 4-carbon. 6 3s are 18. 1 6 are 6. 18 plus 6 is 24. 6 4s are 24. First stable product is a 4-carbon compound. That is the reason it is called C4 cycle. Where this reaction occur, not in bundle sheath, mesophyll, it occur in mesophyll cells. This reaction occur in mesophyll cells. What will happen to 6 molecules of oxaloacetic acid? It may be transaminated to form 6 molecules of aspartic acid. It is also a 4-carbon amino acid. It's an acidic amino acid. R is CH2COOH is aspartic. CH2COOH is glutamic. You studied under biomolecules. So oxaloacetic acid can be transaminated to aspartic acid. Okay, leave it. It's not needed. It's not needed for us. 6 molecules of oxaloacetic acid forms 6 molecules of malic acid. Naturally, this is the reduction process. I don't want to talk all that. This is also a 4-carbon compound. Okay, up to here no problem. First, PEP is absorbing CO2 and form oxaloacetic acid. Oxaloacetic acid is forming malic acid. What happens next? What happens next? This malic acid through plasmodesmator is transported to bundle sheath cells. Yes, bundle sheath cells. So a 4-carbon organic acid through plasmodesmator. Why only plasmodesmator? Because they are suberized. I told you, bundle sheath cells are suberin. Suberin is impermeable to water. So it forms malic acid. What will happen to the 6 molecules of malic acid? It forms 6 molecules of pyruvic acid. And pyruvic acid is a dash carbon, 3 carbon. This is dash carbon, 4 carbon. 6 4s are 24. 6 3s are 18. There is a loss of 6 carbons. That is in the form of 6 CO2. Yes or no? And I told you, this reaction occur in which cells? Bundle sheath cells. Is Rubisco there in bundle sheath cells? Yes. Can Rubisco, listen carefully, can Rubisco in bundle sheath cells get oxygen? No. Why? Because walls are suberized. Suberin is impermeable to water. Cork, bottle caps are made up of cork. Have you ever seen? Because it prevents the entry of moisture from outside to inside, because cork is made up of suberin. So Rubisco site, if I'm an enzyme, Rubisco, one side is for RUBP, other side is either for CO2 or oxygen. No way oxygen can reach because the entire walls are suberized. So can photorespiration occur in C4 plants? Nahi. Then can Calvin cycle occur in C4 plants, bundle sheath cells? Yes. How? Is CO2 passing through suberized walls? No. If oxygen cannot enter, even CO2 also cannot enter, then how can Calvin cycle occur? Malic acid runs through plasmodesmator, carrying this carbon dioxide into bundle sheath cell and liberate that CO2 to form pyruvic acid, and that CO2 is accepted by RUBP. This is accepted by 6 molecules of RUBP to form 12 molecules of PGA. It forms 12 molecules of PGL. 10 and 2. These 2 molecules form glucose. These 10 molecules. Somewhere we have seen this. Yes, children, you recollected correctly. You are very smart. And these reactions are nothing but Calvin cycle. Where is Calvin cycle occurring? Bundle sheath cells. Why? Because bundle sheath cells are properly suberized and Rubisco site is never exposed to oxygen. Can dark phase occur in bundle sheath? Yes, Calvin cycle occur in bundle sheath. All right, then this pyruvic acid, its job is over. Now, in the presence of ATP, 12 ATP molecules form phosphoenol pyruvic acid. You got a doubt? 6 molecules by 12. In order to add 6 inorganic phosphates, in order to add 1 inorganic phosphate to 1 pyruvic acid, I require energy of 2 ATP molecule breakdown. ATP here is acting as energy donor, not phosphate donor. Did all of you understood this? 2 molecules of ATP first break, liberate energy, and that energy add 1 inorganic phosphate to pyruvic acid to form phosphoenol pyruvic acid. Finished. How many ATP needed here? 12 here, 18 here. Total 30 ATP are needed in the entire C4 pathway. Now I'm pretty sure that all of you understood this C4 pathway. Look at this. Atmospheric carbon dioxide accepted by PEP forms a C4 acid. क्या है वो C4 acid? Malic acid. First oxaloacetic acid. Oxaloacetic acid, you say first stable product, but I'm sorry to say that it is not stable. The most stable form is malic acid. That is the reason oxaloacetic acid is not transported. Instead, malic acid is transported to bundle sheath cells. All right, this malic acid which is transported. But if they ask you what is the first stable product in C4 pathway, for God's sake, don't tick malic acid. You tick oxaloacetic acid only. But when compared to oxaloacetic acid, malic acid is more stable. That is the reason this C4 malic acid is transported to bundle sheath. It undergoes decarboxylation. Again to form what is called, and that CO2 lost is accepted by RUBP and Calvin cycle. Look here, Calvin cycle. And this C3 acid, what is this C3 acid? Pyruvic acid. It's transported back into mesophyll and it is used to form again phosphoenol pyruvic acid. Easy way to remember. Just shall I tell you the easy way to remember? This is PEP. This is forming oxaloacetic acid. This is forming malic acid. Malic acid is transported and it is forming pyruvic acid. Pyruvic acid is transported back. This is it. And this CO2 lost here is accepted. This is Calvin cycle. So all these reactions are in fact known to you. This is 3 carbon. This is 4 carbon. This is 4 carbon. This is 4 carbon. This is 3 carbon. This is 3. So 3 carbon organic acid in this direction. 4 carbon organic acid from mesophyll to bundle sheath cells. Nice. I'm getting exhausted here. All right. But I don't think you're getting exhausted because you're all very smart students. So you can easily understand what all I'm saying. So we are winding up, children. Last 5 minutes. All right. Just we'll see about C4 pathway. Haslak pathway. Definition. It's an alternative pathway of carbon dioxide fixation. Occurrence in tropical SMS. Sugarcane, maize and sorghum. Is it seen in India more or America? Mostly India. India is what? Subtropical. So therefore, tropical or subtropical is sugarcane, maize and sorghum. And in tropical regions, temperature is more? Yes. Carbon dioxide saturation is 360 ppm. Atmosphere, 360 ppm. Saturation means highest photosynthetic activity, 360 ppm. So it means that the present level of carbon dioxide in the atmosphere is enough for a C4 plant to reach saturation. You need not enhance CO2, or you need not use a chopper to fumigate the fields with CO2, or in advanced countries we do it, or grow them in what is called greenhouses. It's not needed. Common example, sugarcane, maize and sorghum. Initial carbon dioxide acceptor is PEP. First stable product, oxaloacetic acid. Operate in mesophyll and bundle sheath. Primary carboxylation, cytoplasm of mesophyll. Secondary carboxylation, stroma of bundle sheath. Remember that. PP carboxylase in cytoplasm. Rubisco, bundle sheath. Rubisco act as carboxylase only. Photorespiration is absent, productivity is high. Crans anatomy, the bundle sheath cells in the form of many rings. This is called crans. It's a German word. So one more language, all right, you know now, that is German. Crans means a group of bundle sheath cells in many rings. All right, arranged in many rings. The bundle sheath cells are large, their suberized walls contain more number of chloroplasts. PS2 is absent. Interesting. Why PS2 is absent? If PS2 is there, there is photolysis of water. If photolysis of water is there, oxygen release will be there. Bundle sheath cells does not want any oxygen. That is the reason they are suberized. So if PS2 is there in bundle sheath, there is a chance for oxygen release. That is the reason even the abandoned PS2, they are agranal chloroplast. Thylakoids doesn't stack to form grana in bundle sheath. So chloroplast of mesophyll cell, grana is seen, Calvin cycle enzymes are absent. Chloroplast of bundle sheath cell, grana is absent but contain the Calvin cycle enzymes. Means within the same plant, mesophyll cell and bundle sheath cell are differing in chloroplast. Dimorphism is seen in C4 plants. PP, CO2, oxaloacetic acid, malic acid is transported, CO2, Calvin cycle, pyruvic acid is transported back. Bondandi. So nicely you explain all this. All right, that's about C4 pathway. I don't think all these reactions are needed. 6 PEP, 6 CO2, 6 oxaloacetic acid. Remember only one point, it occurs in cytoplasm of mesophyll. First stable product is this. And the 6 oxaloacetic acid will be converted to 6 molecules of malic acid. All right, malic acid or aspartic acid, either of them can enter the bundle sheath cells. Then this reaction is important. 6 molecules of malic acid is converted to 6 molecules of pyruvic acid. Decarboxylation. Carbon dioxide lost acted by RUBP. Calvin cycle operate. Decarboxylation and carboxylation, both of them occur in bundle sheath cells. Alright, decarboxylation is a prerequisite, all right, for Calvin cycle to occur. So that's what it is. Secondary carboxylation occur in bundle sheath cells. Rubisco site is never exposed to oxygen. Rubisco site is always exposed to CO2. No chance for photorespiration. 18 ATP and 12 NADPH2 in bundle sheath. Another 12 ATP in mesophyll, total 30 ATP. All right, this is the 30 ATP. So now differences between C3 and C4. RUB primary electron acceptor, C3, this is C3, this is C3, this is C4, okay? Primary carbon dioxide acceptor, RUBP, and then C4, PEP. First stable product, oxaloacetic, first stable product, PGA, C3, oxaloacetic acid, C4. Type of cells involved, only mesophyll, C3, mesophyll and bundle sheath, C4. Right, next, Calvin cycle enzymes are present in mesophyll cells in C3, bundle sheath cells in C4. Starch is synthesized in mesophyll cells in C3, bundle sheath cells in C4. Photorespiration is seen in C3, can never be seen because rubisco is always exposed to CO2, C4. Productivity less in C3 because of photorespiration. Productivity is more in C4 because there is no photorespiration. Oxygen play an inhibitory role, C3. Oxygen can never play an inhibitory role, C4. Only one type of cell, mesophyll, C3. Mesophyll and bundle sheath, C4. Cran's anatomy is absent, C3. Present, C4. Chloroplast dimorphism is absent, C3. Chloroplast dimorphism is present, C4. Tomato, bell pepper, chlorella, C3. Majority of plants are C3. SMS. No, not the one what you're thinking. Sugarcane, maize and sorghum is SMS. That is C4. All right. Temperate plants is C3. Tropical plants are C4. Sugarcane, tropical, easy way. Low temperature optima, 20 to 25 degrees is C3. 30 to 40 degrees is for C4 because they are tropical. CO2 saturation for a C3 plant is 450. Don't think C3 350. No, C3 450 and 4 350. All right, be careful about this. That's all, Indy. That's about the differences between C3 and C4. You are extremely good in remembering all these points. That is very, very simple. Now, we are about to wind up now. What is the last part? Factors affecting photosynthesis. All right. Now, when we talk about the factors affecting photosynthesis, children, law of limiting factors, the scientist's name is Blackman. What is the meaning of limiting factor? Any factor which is less than what is needed is said to be a limiting factor. All right. Process is controlled by suboptimum factor. That is a limiting factor. Now, you know what Blackman said. Let me use in his own words. If a process is conditioned its rapidity by several factors simultaneously, then the rate of the process is controlled by the pace of the slowest factor. No, this is very complicated. Shall I make it simple, children? I'll do it. That is the reason I'm here. Now, if a process like photosynthesis is controlled by several external factors, because internal factors never act as limiting. If several external factors simultaneously, like CO2, light, temperature, water, then the rate of photosynthesis is decided or controlled by the factor which is present in less than the optimum quantities. Did you understand what I said? That factor is said to be suboptimum factor. Look here. This is 0%, this is 5% of sunlight, this is 10% of sunlight, this is 15%, and this is 20% of total sunlight. Look here, as long as sunlight is in less than 10%, look here, the rate of photosynthesis is increasing. Then look here, at 10% of total sunlight, all right, saturation is reached. Beyond that, even if I increase the light intensity, there is no increase. So light saturation can be reached at 10%. So 10% of sunlight is enough to reach maximum saturation ante maximum photosynthetic rate. So look here, light is acting as a limiting factor as long as it is less than what is needed. Yes or no? Less than optimum. That is said to be a limiting factor. So shall I say which act as limiting factor here? The x-axis is light. Here light. Here light. Here light. Because all this is suboptimum. 10% is saturation. Suboptimum. Here not light. Here not light. Here not light. Because it is more than optimum. So at this point, at B and at C, at B and C, it could be CO2, which most common limiting factor is CO2, or it could be temperature, or it could be any other external factor. Did all of you understood this? So this is said to be law of limiting factors. Most common limiting factor is carbon dioxide. Internal factors will never act as limiting factor. Nice, I explained nicely. All right, now at low light intensity, C3 and C4 plants respond to high CO2 concentration At low light intensities, neither C3 and C4 plants respond naturally. At low light intensity, there is no ATP and ADPH2. There is no synthesis of ATP and ADPH2. There is no dark phase. Light saturation is reached at 10%. We already discussed. Red light and blue light, this is 10% saturation. Beyond 10%, there is no further rise. Most common limiting factor, CO2, we already discussed. C3 plants, 450. CO2 saturation, 350. What is parts per million? I told you, 350 carbon dioxide unit. Carbon dioxide molecules are present for every 10 power 6 air molecules. That is the most common, what is called carbon dioxide concentration in the atmosphere. And CO2 saturation in C3 plants is 450. At 450 saturation is reached, means you have to increase this. And that is what greenhouse crops is, enhanced CO2. Temperature has a role both on light phase and dark phase because enzymes are involved. What are the two enzymes in light phase? FNR, ferredoxin and ADP reductase, and one more is ATP synthase, coupling factor. Water has more a role on plant rather than on photosynthesis. More water, guard cells become turgid, stoma opens, more uptake of CO2, more photosynthesis. Same way, more water, leaf cells elongate, more surface area of leaf, more uptake of CO2, more photosynthesis. That's all. That completes a chapter called photosynthesis. I said 1 hour, but it took a little bit more time, but no problem. I'm pretty sure that you are extremely good in revising this topic. And thank you very much. We are going to meet with one more topic, respiration, in tomorrow's class.